All right, good afternoon, everybody. My name is Ben Augenbraun. I'm a member of the chemistry department, but I also serve on the lecture committee, who is putting on this series of faculty lectures. This is the third in a series of 6 that are continuing until spring break. And one thing I did want to share, just a scheduling update, is that next week's talk is Professor Ralph Morrison from math, and that talk is actually going to be in Wege Auditorium rather than here. So Wege across the street in the chemistry building. So like I said, today is the third of 6 exciting talks where we've invited faculty to share their fascinating scholarship. Today you'll be hearing from Professor Bob Rawle, who is an associate professor in the Department of Chemistry. He is a biophysical chemist by training and studies the biophysics of viral infection and lipid membranes. He received his bachelor's degree in chemistry from Pomona College in 2008, where he worked in the laboratories of Professors Cynthia, Selassie, and Malkiat, Johal, to develop bio-analytical methods to study DNA damage. He then earned his PhD in chemistry with a focus on biophysical chemistry from Stanford University in 2014, working with Professor Steve Boxer, studying the membrane biophysics of vesicle fusion. Followed that up with postdoctoral work at the University of Virginia Medical School, where he worked with Professor Peter Kasson, studying the biophysics of influenza virus and Zika virus membrane fusion. And afterward, he had a short, but he says awesome stint as a stay-at-home dad. So Bob wrote that he loves teaching both inside and outside of the classroom. At Williams, he teaches classes in introductory chemistry, biochemistry, and biophysical chemistry. On a personal note, I've been very fortunate to work with Bob closely as we've taught parallel sections of our department's introductory class. And I'm truly grateful to have learned so much from him about teaching, especially through the enthusiasm and humor that he brings into his classroom. And I'm so happy that all of you are going to get to experience that right now in Bob's talk. I assume. So please join me in giving a very warm welcome to Professor Bob Rawle.
All right, thank you. Thank you. I'll send you your payment afterwards. Wow, it is great to see all of you. Some of you I know, others I don't. Welcome. I'm happy to be here to talk with you today about viruses. I put this backdrop here, this really cool image developed by this set of scientists at Visual Science of, it's a structural model zoomed in of SARS-CoV-2, which is the virus that causes COVID-19. I'm actually not going to talk much about this virus, but I put it up, A, because it's a cool graphic, B, because I think it highlights to us that we need to understand viruses better. This became very apparent to all of us during the global pandemic. And so today, my goal is to share a little bit kind of big picture knowledge about viruses, how they operate, what they are, and then a little appetizer of the types of work that we do in my lab and how we go about doing that. So that's kind of where we're headed today. Before I begin, I wanted to do 2 things. The first is to tell you a little bit about just like who I am as a researcher and what are the types of work that we do in my lab, because I'm only going to talk about a small fraction of it today. It's captured well, I think, by our lab logo. So we're biophysical chemists. We study viruses. That's the head here. The little stick figure guy is surfing on a microscope cover slip because we do a lot of microscopy in our lab. We think quite a lot about lipid membranes, and this little squiggle is meant to represent a lipid, one of the key components of a membrane. And we are in a chemistry department, which is why it's holding a beaker. The types of stuff that we do are not what you would traditionally think of as chemistry, but belong very well in a chemistry department. So part of my job today will be to talk a little bit about that. The other thing that I want to mention up top is to give a plug to all of the awesome students that I've worked with in my lab over the years. So in the corner of my lab, there is this wall where we have a picture of everybody who's worked in the lab. And the work that we do, it would not be possible without them. So much of the stuff that you're doing, it involves lots of heavy lifting, work in late nights at strange times by people in my research lab. So I'll be highlighting them as we go along. But part of what I wanted to say is that this is what brought me to Williams in the first place, is that I wanted a place where I could invest in teaching and be rewarded for that. but also do good scientific research and train students in the process. And Williams allows me to do that. I'm really thankful for the support that Williams has given to the lab sciences in general over the years, and in this climate of uncertain funding in the future for our country. I hope that that continues to be an institutional priority. So I just wanted to say that up top. Alrighty, where are we headed today? First, I'm going to give you a little bit of an overview of what virology is and what viruses are and how they operate. I warn you, this is going to be like half the talk because I think it's, A, you probably don't think as much about viruses as I do, but B, there's a lot of misconceptions about viruses, and I think it's important as an educated academic audience that we have a general sense of what they are and how they operate. So that's going to be the first portion of the talk. The second portion is going to be giving you a little appetizer of the types of things that we do in my research lab. We're just going to talk about a couple questions that we've been looking at. I'm more interested that you understand kind of our approach, because the approach is part of what makes the science interesting to us. Alrighty. So let's start at the very beginning. What are viruses? As with many things in academics, defining something precisely is not always so easy, and viruses are no exception. This is my take on it. Viruses are obligate intracellular parasites. That's a mouthful. Obligate meaning they're required to replicate themselves. Intracellular meaning inside another host cell. And parasite means they steal the resources of that host cell to replicate themselves. That's kind of the big picture idea. Even that is still, I think, kind of a little bit more complicated than my brain wants to handle it. So I often think of them as molecular machines. So they are machines that are designed to replicate themselves inside some host. And that's really all that they are. There's nothing more magical to it than that. Over here on the right is a scanning electron micrograph of a cell. So the thing that is in red is some poor cell that is trying to be infected by all of these little yellow dots, which are SARS-CoV-2 viruses. And one of the things that I want to emphasize is what viruses are not. They are not sentient. That seems really silly to say, but you'll often hear them talked about in a way that makes them seem like they're sentient, like they want to do something, or they're sneakily evading our vaccine strategies or things like that. They're just dumb machines. They're dumb old molecular machines. They're bouncing around in their environment until they find the right conditions that trigger them to initiate replication. And what I view my job and the people in my lab and the people in the community, the scientific community that I'm involved with, our job is to understand the machine, and then once we understand it, sometimes use it for our own ends or break it is more often the case. What we do in my research lab is mostly trying to understand the machine and how it works. Are they alive? Oh boy, that's a separate question. You can ask somebody from philosophy is talking in a little bit. You can ask them about that. Okay, next point. Viruses are everywhere. If you swab any surface in this room, you'll find evidence of viruses. You are all breathing viruses in and out right now, I guarantee it. That makes us all a little bit germophobic, but it is the way that the world exists. Every day you're eating or breathing billions of viral particles. We actually have them as part of our own genetic material. I'm happy to talk about that in questions at the end. It's kind of an interesting fact that we're partially constructed from viruses themselves. They infect every living thing that we know of. Bugs, birds, fish, bacteria. If it's alive, there are viruses that infect it. So they are all around us all the time. How do they make us sick? Well, first they have to infect us. I'm going to talk a lot more about that process here in a little bit. But after they've infected you, there's two main ways that viruses cause disease. The first way is direct cell damage caused by the virus. So after it gets inside and it replicates, that will often damage the cell that it's inside of. And if that cell is important, or if there are enough of those cells that get damaged, then that can cause disease symptoms. So for example, the first time, the only known time that I was infected with SARS-CoV-2, I ended up getting like some nerve damage in my face. So I have like now a little bit of partial numbness on one half of my face. No tests were run. I don't know the details for sure, but probably that was the virus got into that nerve cell, damaged it, and it was direct cell damage. And so that's an example of like a type of disease state that a virus could cause. The more common types of disease states that occur from viruses are actually damage that we cause to ourselves. So in the process of our immune system trying to get rid of the virus, it'll often destroy our own cells as collateral damage, and it will also cause systemic effects. So this is one of the reasons why many viruses give you all the same set of symptoms, right? Many viruses give you fever and runny nose and body aches and maybe some GI symptoms. And that's because those symptoms are caused by your immune system, not by the virus. It's the response that actually causes more damage in some instances. Amazingly, we are not sick all of the time. And there's different reasons for that. One is that for most viruses, we're actually not very good hosts for them. So they don't infect us very well, or if they do infect us, they don't cause disease. There's a good fraction of viruses that can replicate in you, and you're none the wiser because they don't cause any disease states. One of the big reasons why is that we have very good immune systems. This is not what your immune system looks like. It's kind of a cool graphic to represent that. But our immune systems are very, very, very good. So even though we're eating and breathing viruses all the time, most all the time we're not getting sick. Next big question, where do viruses come from? This is a big question and not very well understood, and probably dates back to the origin of life. So in the earliest evidences of life, there's evidence of viruses. So I'm actually not going to answer this question. I'm going to answer a related question. Let's assume a virus exists already. How does it get into people? And the answer is through animals. So this is what's called a zoonotic spillover. This is a graphic that was taken from a review article that shows the likely origins of different coronaviruses and how they get into people. And so for SARS-CoV-2, the strongest evidence is that it started off in a bat, and then it probably went through some intermediate animal. Here they were wondering if maybe it was a pangolin before it ended up getting into a person. And the way that works is the person is eating the animal or working with the animal or somehow has contact with the animal. They get the virus from the animal, it gets into the person, and then it begins to spread among people. These sorts of spillover events, they happen all the time. Anybody who's working with animals is going to be exposed to viruses from those animals, and they'll often get infections from those animals. What makes something a sort of pandemic potential virus or a dangerous virus is that after it gets into person A, it then mutates enough that then it can spread quickly among other people. And that usually is the biggest barrier for pandemic potential viruses. Unfortunately, we just all have been living through the SARS-CoV-2 pandemic. It's going to happen again. Like this is not bump this microphone. It's something that will happen again, given the global nature of our economy and the interactions that we have with wildlife and other animals. It for sure is going to happen again. You probably have heard in the news about the H5N1 influenza virus that has gotten from birds into cows and into cats. And there's some people that have been infected with it. That may or may not turn into a pandemic virus. It's hard to predict these sorts of things, but it will happen again in the future. This underscores the need to really understand these viruses and be able to come up with strategies to defend ourselves against them. What do viruses look like? They come in all sorts of shapes and sizes, and these schematics sort of represent idealized pictures of what some particular viruses might look like. This is an electron microscopy image that was taken here at Williams by a student in my lab, Kirky, a few years ago, of Sendai virus. This is the virus that I'm going to talk about in the second half of the talk. And so if you squint very roughly, you can kind of see the key features of this idealized picture of the schematic. All viruses at their heart have genetic material. So that genetic material contains all of the information about how to produce a new virus. So it's this little squiggly that's here inside Sendai virus. For influenza virus, it actually comes in different pieces. So all these different little squigglies would be its genetic material. Adenovirus, bacteriophage, they're going to be inside these little capsules. And this could be RNA or DNA. Different viruses operate in different ways. But if you think kind of as a kind of biophysical chemist, as an engineer, as a structural person, how a virus is organized is its most important information is in the middle, And then there's a structural housing around the outside that protects it. And that structural housing is designed to protect it, but also to shuttle it between one host and the next. So everything that's on the outside here needs to have the ability to transfer the virus from one host to the next. Many viruses are part of this structural housing includes a lipid membrane. So that's true for Sendai virus, it's true for influenza virus. Some are conducted only of protein coats, so they're very highly organized proteins. The viruses that I study all involve lipid membranes, and these little blobs that are pointing out of them are going to be proteins that are designed to do specific things, and I'll talk about examples of that here in a minute. How big are they? They're usually really small, about 100 nanometers. That size dimension probably doesn't mean things unless you think on the nanometer scale a lot. It's like 1,000th the width of a human hair, super, super tiny, are generally considered would be most viruses. Okay, so now let's talk to the thing that I really want to get to here, which is the infectious cycle or the life cycle of a virus. How do viruses infect host cells? This is a huge question, and depending on the virus, the answers may or may not be known very well. So some they know lots of, some are very little. In general, there's broad categories that you can break down, and the particulars vary from virus to virus, but the general categories are going to be the same. This is meant to represent Sendai virus. I'm going to sort of introduce it in the context of the virus that we study, or one of the viruses we study a lot in my research lab. Down here is sort of a simplified version, a chemist's version of a cell, where this is the outer cell membrane. This represents the nucleus. One of the big points I want to make about this, and pretty much everything that I talk about, is that it's a dramatic oversimplification. And that's emphasized by this little inset over here. So this is an artist's rendition of what it would look like inside a cell if you drew things according to the size and number density of all the objects that would be there. And what you can see is that it's very highly packed and very chaotic. There's lots of things going on inside here. But when we especially as biophysical chemists, but often just as scientists in general, think about problems, we often are trying to reduce them down to the simplest components that we have control over to kind of make sense of the system. And so going forward, we're going to totally ignore this complexity here and be thinking about a very simple system, but we have to remember that it does exist underlying everything that we're talking about. To emphasize that, I'm going to change the title here to dramatically oversimplified model of the Sendai virus life cycle. And any of you that have been my students will recognize that I say that all the time. Okay, so with that, let's begin. So the virus is a dumb old molecular machine. It's bouncing around in its environment until it encounters something that will trigger it to initiate replication. This represents some cell that's susceptible to infection. Eventually, if the virus bounces close enough to the cell membrane, and if that cell membrane has particular molecules on it that the virus can recognize, then it will bind to the surface of the cell. Those molecules are called viral receptors. They could be a particular protein, they could be a particular sugar, they could be a particular fat molecule. There's all sorts of different things that they could be. But the virus will have evolved the ability to recognize that particular molecule on the surface of a cell so that it will know that, no, I'm using it in, it obviously doesn't know anything, but so it will recognize sort of through chemical means that this is a cell that would be a good host for it to replicate inside of. This binding step is very important because it's one of the determinants of why it helps explain why viruses infect certain tissues and certain organisms. So respiratory viruses, like SARS-CoV-2, like influenza, they have evolved the ability to bind to molecules that are expressed on respiratory epithelial cells. So that's one of the reasons why they infect our lungs. Viruses that will infect a bat or a horse or a pig will have evolved the ability to recognize molecules inside those organisms. This is not the only determinant of why viruses spread between different tissues or different organisms, but it's one of the key components. So understanding this binding step is very important. The next step is what would be called membrane fusion. This would be true for viruses that have membranes around them. The idea is that in order for the virus to replicate, it needs to get its genetic information inside the host cell, and that requires it to open up a hole in between the structural housing of the virus and the membrane of the cell. This picture over here shows a kind of better schematic view of what this might look like. The idea is that there's particular proteins on the virus whose job is to basically open up a hole inside the cell membrane and allow the genetic material to then get inside. The next step is replication. So the main idea here is that the cell on its own will be replicating its own genetic material, making different things. And the virus confuses the cell and makes it think that the genetic material that it has injected is actually the cell's own genetic material. And so it will begin replicating that genetic material and it will produce the products that the information in that genetic material tells it to make. Those products could be more copies of the genetic material, so that's all these things down here. It'll be more copies of the structural housing, so it'll tell the cell to start producing viral proteins and more components of the lipid membrane up here. And then there will be a whole host of other proteins that do other things. It'll help it evade the immune system, it'll help it replicate the viral genetic material, that sort of thing. One of the big ideas is that when the genetic material is produced, it will happen with errors. So for a virus that is composed of RNA, like Sendai or like SARS-CoV-2, you're going to get about one error every time you replicate the virus. So in some ways, it's kind of bad at replicating itself. But that's an intentional strategy because that allows it to evolve very quickly. So you're producing many, many progeny, lots of different errors in the different progeny, and some of those will be beneficial for the virus to survive under certain conditions. Alrighty, the next step is assembly. There's a lot of interesting biophysics that happens here, but the big picture is that each of these components that are produced will have some sort of affinity for each other. They're kind of like magnets that can attract in a way. And so what will happen is that these proteins that are produced will cluster together. They will recruit other proteins next to them. The genetic material will also end up sticking to the same surface there. And this is all through random chance. This is not like they're directed and they're finding each other out or shouting some signal. They're just dumb old molecular machines. They're bouncing around in their environment until eventually they find something that they stick to, and they're more likely to stick to the things that make up themselves. So they're all going to end up clustering here at the cell membrane, and eventually that clustering will cause the membrane to start to bud outward, and then it will pinch off, and that will release one virus out into the space outside the cell. That process happens many, many times. So for a single virus that goes in and infects one cell, you're going to get many viruses that come out the other side and are reproduced. These can then go on and infect the neighboring cell, and then the replication cycle begins again. So then how does it get into new organisms? It's kind of as you might imagine. So let's talk about a respiratory virus. Eventually, the virus has infected enough cells inside your lungs, and it's produced enough virus that as I'm talking, I start producing little droplets that have virus particles in them, and they go throughout the air and get into somebody else's lungs. So if I have enough virus, and if the particles are produced under the right conditions, and if there's enough viruses in them, and if enough viruses survive the process to end up in somebody else's lung, and if that person's lung has the right receptor, and if it evades the host immune defenses, then maybe it could infect the next person, right? So there were lots of ifs in that statement. There are many barriers that could cause the virus to not sustain an actual infection. It seems like it would be very unlikely to occur, and it is. It is very unlikely. But with viruses, their strategy is all about the numbers. So even if something has a 0.001% chance of occurring, if it's producing 10 to the 8th viral particles, then actually the odds become pretty good that the infection can occur from one individual to the next. And that really is the strategy for viruses. I only put up here schematics of respiratory transmission. There are other transmissions, sexual transmission, fecal oral, blood contact. It would be a more risque talk if I put those up. So we're just going to stick with respiratory for right now. Finally, let's talk about drugs. How do antiviral drugs work? The high-level idea is pretty simple. If you can understand the infectious cycle, a drug is a molecule that blocks one of those steps. And that really is the key idea of any drug molecule. So this is showing an example for SARS-CoV-2 again. And different people have developed or tried to develop molecules that block different steps in the process. So if you block binding, then the virus can't get in. If you block fusion, then it can't release its genetic material. There's inhibitors or drugs that block particular steps in the replication cycle once it gets inside. People, not for SARS-CoV-2, but have developed drugs that kind of work on the outside. Anywhere along the process, if you can stop it, then you have developed a good drug. You may have heard of Paxlovid. Paxlovid is what's called a protease inhibitor. As the genetic material gets produced, it needs to be processed in a certain way, and it basically blocks that step. And that's how Paxlovid works to reduce the effects of SARS-CoV-2 infection. Remdesivir is another one you maybe have heard of. If you can't take Paxlovid, you take remdesivir. That one is, it blocks the replication of the genetic material specifically. And that's basically how it works. So this highlights the need, if we understand the process of viral infection, we understand how the machine works, then we can figure out ways to try to stop it. In my research lab, we focus mostly on the binding and the fusion steps. And we're not really in the business of developing drugs against them, although we do collaborate with people who do. We're really trying to understand the machine as kind of the first step. Alrighty, so that's the general virology section. If you're interested in learning more, you can check out these awesome resources. There's this great online virology course from a professor at Columbia. Goes into lots more detail about all these steps if this is something that's interesting to you. Or if you're a real nerd like me, you can listen to the This Week in Virology podcast, where they go over all the hot research that's come out recently. They actually do a very good job of breaking it down for a more general audience. So it's a good listen if you are interested in that. Alrighty, so that was our general kind of virology background. Now let me talk about a couple questions of things that we look at in my lab. I'll be talking about Sendai virus. I would wager that most of you have not heard a lot about Sendai virus, unless you work in the animal care facility, and then you get emails from me every 6 months or so. I'll talk a little bit more about that. So the reason why is Sendai virus, it belongs to a family of viruses called the paramyxoviruses, which cause a lot of respiratory diseases. So you may recognize parainfluenza, measles, mumps. This is kind of a genetic family tree, so the closer they are together, the more related they are. Sendai virus is very closely related to viruses that cause quite a bit of respiratory infection in humans. But Sendai virus itself, it can infect human cells. It only causes disease in animals. So this is a convenient candidate for us to work with because of biosafety considerations, which I'll talk about a little bit more here in a second. But also because if it can infect human cells but not cause disease in humans, then this makes it a very good biotechnology tool, because you could then use it to deliver things that you might want. So this virus has been used as a candidate for vaccine development, for gene therapy deliveries, all sorts of cool biotechnology applications. Our goal is to try to understand how it works, partly as kind of a representative of this family, to kind of get a window into how this family works, but also to help understand how we can make it a better tool. Let me talk a little bit about biosafety. I think this is important, again, for people to kind of understand how we operate with viruses here at Williams and more broadly. So at any academic institution or any research institution where you're conducting research with potentially pathogenic organisms or viruses, there's an entire regulatory committee that's responsible for overseeing every single experiment that you do. This is called a biosafety committee. That biosafety committee will try to assess what is the risk level of a particular virus, what's a risk level of the experiment, is there a danger of release into the broader public, is there a danger to the experimenter, How's the virus stored? What are all the steps of every experiment? It goes into very nitty-gritty detail about everything. To help in that process, pathogens are usually categorized by a series of biosafety levels. And so these are the broad categories. I'll talk just briefly about each of them. Biosafety level 1 are agents that are not pathogenic at all. You can just handle them on a regular benchtop. Certain strains of bacteria are this way. Most of the ones that if you take a biology class or a biochemistry class at Williams and you work with a bacteria, that will usually be a very non-pathogenic bacteria. It would be BSL-1. You don't really need any precautions for that. Biosafety level 2 are ones that have a moderate risk level. What does moderate mean? Moderate means that there's maybe the potential for mild disease in the experimenter, that there's little potential for release into a larger environment, or there might be more moderate disease in the experimenter, but the odds that it would be contracted in a laboratory setting are very low, and that would be considered kind of a moderate risk level. Sendai virus is classified at Williams as a BSL-2. It doesn't cause disease in humans, but it can infect human cells, and so if you're immunocompromised, you need to be extra careful about it. And more importantly for us, it can cause disease in animals, so if it gets into the animal care facility, that's a big problem for anybody who does research with our animals. And so we have to take a bunch of special precautions, and I'm happy to talk more about that in the questions if you'd like to talk about that, to make sure that none of that happens. This is the highest level that would be approved at Williams. So we would not have anything that would be above that level that would be occurring in research here at Williams. In case you're interested, biosafety level 3 now is serious risk level. This usually means that there's a considerable risk for the experimenter, but moderate risk for it getting out and being transmitted more broadly. But it requires a lot of special facilities. So you're working with respirators. Usually you're working in a negative pressure room so that no air will escape that might have the pathogen inside it and accidentally infect somebody in the hallway. Something like certain strains of tuberculosis would be considered this. Biosafety level 4 are viruses that are not well characterized. Usually they're emerging viruses. There's usually high risk to the individual and to the wider community. They require like entire separate buildings. These are ones like if you see a movie and somebody's working with a virus and they're like in a spacesuit, this is the level that it's working with. So like Ebola virus would be an example of something that would be conducted in one of those facilities. There's only a handful of these facilities in our entire country, or even more broadly in the entire world. So there's only set locations that those would work like that would even be allowed to be pursued. Anyhow, that's a little bit of a tangent, but I think it's important, again, as an educated academic audience, that we kind of understand how these things kind of play out in a research setting. Alrighty, back to Sendai virus. In my lab, like I say, we're really interested in studying the binding and the fusion step. Today I'm just going to talk about questions relating to binding, although I'm happy to talk in questions more about fusion. The key players have been identified for this virus. So you can see that these different proteins have been identified. This HN protein is the protein that's responsible for binding to the receptor on the host cell membrane. This receptor is a sugar that's attached either to a protein or a lipid. It's a particular type of sugar called a sialic acid. And the HN protein has evolved the ability to recognize that and bind to it. One of the big ideas that I want you to know right now in terms of general background is that in order for the virus to remain stably bound to the surface, it needs to bind to many receptors at once. So just binding to one of them is not going to work. You have to bind to a whole bunch of things all at the same time in order to actually stay stably bound. And that'll be important for things that I'll talk about here in a minute. How are we going to study binding? Our approach is very much a biophysical approach. And so what do I mean by that? We try to distill things down to very simple components that we have a lot of control over so we can ask very targeted questions. The approach that we have taken is we are fluorescently labeling our virus and looking at it on a microscope. It's all conducted in this little reaction chamber called a microfluidic device where we can control the volume very, very precisely. And one of the key ideas that we do is instead of watching the virus try to infect a cell, we watch it try to infect a membrane that we have created ourselves. These are called model lipid membranes. And the reason for that is we gain a lot of control. We can do things with these membranes that we could never hope to do with a cell membrane, and I'll show you an example of that here in a little bit. The price that we pay is we lose a lot of complexity. So the complexity, the cell membranes are much more complicated than the things that we work with. So we have this kind of trade-off in terms of control versus complexity. I'm going to talk about how each of these pieces work so you can understand the experiments that we do. Let me first highlight, this is the team of students a number of years ago that really helped get this system up and running. It's a very cool system. Each one of these little individual dots is a single virus that we can observe on our microscope. We can track it. We can do all sorts of cool stuff with it. They were really instrumental in getting it up and running. Today, I'm mostly going to be talking about questions that Amy was looking at, but they all did awesome work in helping to get this system up and going. Okay, so let me talk a little bit about each of these pieces so you can kind of make sense of the measurements that we do. The first is this microfluidic device. I actually have a little copy of one here, so I'm going to hand it around so you can look at it. There's no virus in here. Instead, what I've done is I put a solution with food coloring inside so you can see the little channels. So I'm going to hand that around, and then I'll start talking about it. Here, yeah. So what it is basically a piece of plastic that we make ourselves inside the lab. And we attach it to a glass cover slip that we could put on a microscope. And inside this little plastic device, there's a channel that's been hollowed out so that the bottom surface is the glass cover slip, and then the top surface is sort of this little channel inside the plastic. We have holes that allow us to use little syringe pumps to precisely control the volume that goes in and out. We do this because the volume that it contains is very small and because we can control the timing and everything very precisely. This is a very simple design that we have here, so it's just kind of like a straight channel. We can actually make them in all sorts of complicated designs. People can do this where you can create pumps and all sorts of things that you can operate as like a little machine inside the device. This one that you're looking at is very quite straightforward and simple. So that's kind of the first piece is our device here. The next piece is how do we make these membranes ourselves inside our microfluidic device. There's a bunch of different types of membranes that we use in our research lab. I'm just going to talk about one type today. It's called a supported lipid bilayer, which is basically a planar membrane that is kind of floating on the surface of this glass cover slip. So how do we do that? To make a supported bilayer, you first need to make some vesicles. Vesicles are spherical membranes that are very small. These ones are about 100 nanometers across. So how do you make a vesicle? Well, you start with lipids or fat molecules that you dissolve in organic solvent. These lipids you can buy from a manufacturer. So companies will go into chickens or eggs or soy or whatever is the source that they are interested in, and they will extract out particular molecules so that you have an isolated set of something that you're starting to work with. So we might mix in cholesterol, which is probably a lipid that you've heard of, and other lipids that you may not have heard of, POPC, which stands for, let's see if I can even remember, phosphatidyl I'm forgetting the last part. You don't even need to remember it because the acronym exists. The idea is that you have these sets of different lipid molecules that are put together in the ratios that we want. And that really is the key idea, that we can mix together the particular lipids in the ratios that we want at the beginning. We then get rid of the solvent that they are dissolved in, and we're left with this lipid film. We then add in water or buffer and resuspend them by mixing them up very vigorously, and that generates these blobs of lipids. If you then pass them back and forth through what's called an extruder membrane, you can end up generating these vesicles. How does that work? There's this extrusion device that basically has extremely tiny holes in it. And so when you pass these lipids back and forth through the holes, they get elongated as they go through the hole, and then they'll kind of pinch themselves off as they go out the other side. And that ends up you do that a bunch of times, that ends up forming these nice spherical membranes on the other side. You can kind of think of it as like you're blowing bubbles, only there's no air and it's underwater, if that makes sense. But the big picture idea is the same, that you're generating something and you're kind of blowing a bubble. Okay, so then once you have these, you can do all sorts of cool things with vesicles. But what we do is if you add them in to one of those devices, so instead of adding in the food coloring, you add in a suspension of these vesicles, they will bind to this glass slide because they have some attraction to the glass slide. And if they bind at a high enough density, they'll actually begin rupturing and merging together laterally. So they all kind of bind and rupture together, and that generates on their own this flat membrane that is attached to the glass surface. So this is called a self-assembly process. So when I say that we make the membranes, it's actually more that they make themselves, and we just kind of give them some conditions to start off with. Given this, now we've talked about this, now you can see how this becomes useful for us in an experiment. Because what I end up with down here, the chemical composition of this, depends on what I start off with back at the beginning that I control. So if I want to do an experiment where I swap the receptors in or out, or I magically remove cholesterol or add cholesterol, that's a very simple experiment for us to do. In a cell, that would be very challenging, and to characterize how well you've done it would also be very challenging. So this is the control that we get is by doing these types of experiments. All right, so then finally let's talk here about how we set up and perform the virus binding measurements. So we have to be able to see the virus. We add in lipid dye. So these are little lipids that have a dye, so it's conveniently I've colored it red. They will automatically sort of partition into the membrane of the virus. It becomes very stained, and that is something that we can then visualize on a microscope. We then basically take a solution of the viruses, we put it into one of those little devices, and then we put the device on a microscope, and we can watch the viruses as they bind to the membranes that we've created. Each of these little dots will represent a virus. We have different computer programs that we have helped write that will track the viruses, count how many are there, pull out whatever is the relevant data that we're interested in capturing for our experiment. Okay, so with all that build-up, I'm going to give you a very quick overview of a couple questions that are the types of things that we can look at in my research lab. It's just going to be a little preview, and I'm happy to talk more later if you're interested in other things that we do. But we'll give you kind of a flavor of like how we could use this to run an actual experiment in our lab. So first, let me talk about a question that we had, which is we know that sialic acid is important for the virus to bind, but are there any other features about the receptor molecule that are important for the virus to bind effectively? So that's a question that we could ask. So this is a question that Amy was looking at. And now that we've talked about how to set up the experiment, you could probably understand in a straightforward way how we could run these sorts of things. So you basically prepare a different membrane that has a different set of receptors. So these were a different set of candidate receptors. We can put them in at exactly the percentage that we want. So in this case, they were all at 2%. And we were basically interested in looking at structural changes, so different chemical structures, and how that might influence the process of the binding. So let me kind of break down these different structures. So this down here, this gray line down below, represents the membrane of the supported lipid bilayer. These would be the tails of the lipid that are pointing into it. And then this stuff on the outside is a sugar. It's kind of hard to see because there's lots of different chemical groups. But these blue things are the sialic acid. So that's one of the pieces of the sugar. And then the stuff that's not colored, in between these three, GM1, GD1A, and GQ1B, they're all exactly the same. So this is the same sugar backbone. And the only thing that differs between these three is the numbers of sialic acids that are attached to them. And then we run an experiment. We see how many viruses bind to the different membranes. So if we look at, here we set GD1A to be equal to 1. We kind of normalized our data. You can see that if you double the amount of sialic acid receptors, that you end up doubling the amount of viruses that bind. So that kind of intuitively makes sense. But the thing that was a little bit interesting and weird was When we looked at GM1, so now we go from 2 down to 1, now we get 0 binding. So it didn't chop it in half, it went down to 0. And the question was, why might that be occurring? There must be something about the rest of this structure that influences the ability of the virus to bind. One of the ideas that we thought of is, well, maybe this extra piece of the sugar, if you don't have this guy up top, it might sort of fold back on itself and prevent access of the virus to this piece over here. And so as a result, it looks like there's one that's available, but actually maybe it's never available. And instead what we're seeing is the difference between 1 and 2 over here, or something like that. So to get at this idea, what we did was we used this molecule that now is just missing the top piece. So all we've done is the bottom 2 are the same, and now we have here. And now, lo and behold, we get half the amount of binding as before. So this seems to suggest that this is blocking access over here, but that once you add in the second one, it actually allows access for this one to begin with. So it's starting to tell you a little bit, again, how the machine operates, that there are these different factors on the host end that can influence how the virus might bind to the surface. So that's a flavor of the types of questions that we're interested in. The other question that I'll talk about, and then I'll wrap up here, is studying the mechanism of viral mobility. So it turns out that after the virus binds to the host cell, it will often move around on the surface to some degree. And we can see that in our own measurements. So this is, well, the coloring doesn't actually help here so much, but this is an image that is overlaid with each of these white spots or viruses, and the little traces are tracking them over time. And so we have a computer program that will go and track where the viruses move over time. And what you can see is, like this red one here, it moves around quite a bit over time. This one that's a blue one moves around a little bit. Some of these other ones are harder to see, but they don't move very much at all. They kind of stay in the same spot. We can characterize the speed at which those viruses move using something that's called a diffusion coefficient. The higher the diffusion coefficient, the faster the virus is moving. And there's a couple things to point out here. You can see there's kind of this long tail, but if you notice, there's a broken y-axis here. So most of the viruses are very slow. They kind of stay in the same spot, or they move just a little tiny bit. But there's a small fraction that move around quite extensively. There's all sorts of cool questions to ask about this mobility. But the question that I want to talk about right now is just like, how do they move to begin with? What is causing the movement of the virus? So the virus is not using energy. There's no little propeller on the virus. How is it moving at all to begin with? And the mechanisms that we were trying to distinguish between were what I would call lateral movement versus rolling movement. So let me talk about these two mechanisms. Lateral movement, the idea is that the viral proteins have bound very stably to the receptors, and the receptors themselves are actually moving around in the membrane. Why are the receptors moving? This is basically the idea of temperature. So molecules are moving around, zipping around at some particular temperature value, and that's going to be true of the molecules in the membrane. So the receptors themselves are jostling about over time. If the virus is attached to stably those receptors, then it's kind of being dragged around as well. That's one idea, is that the virus is kind of just stuck, but it's moving back and forth laterally. The other idea is that maybe the virus is rolling. So maybe these attachments are not very stable, but it's kind of binding and unbinding all the time. But that means that if it binds a little bit more on one side, it might roll on that side. And then if it binds a little bit more over here, then it will roll on this way. And then it will kind of start conducting this random walk around the surface as it rolls and fluctuates which things are bound and which are not bound. So how could we address a question like that? To do this, we ran an experiment looking at the rate of the virus's unbinding from the surface as a function of whether or not we have antibodies present. So let me talk a little bit about how we would do that. The virus, it's attached to our supported lipid bilayer. If we watch them over a long period of time, they begin to unbind from the surface. And we can quantify that. So this is the fraction of viruses that are bound over an hour. And you can see that for this particular experiment that we ran, they kind of go like this. We can fit that to a mathematical decay curve and get a particular characteristic decay time. Then our idea was we would add in some antibodies. These antibodies are proteins that will bind to the viral HN protein. And the antibodies themselves are very big. So if the virus has remained stably bound to the surface, they actually can't fit in this interface and cause any problems. So the virus will stay stably bound until eventually it lets go, and the rate at which the virus unbinds would remain the same with the antibodies present. If, on the other hand, the virus is rolling around, now when it exposes sort of its underbelly, the antibodies can bind and they'll prevent it from rebinding, and so the unbinding rate would become much faster. And that's the basic idea of this experiment. These antibodies were produced in a collaborator lab at Mount Sinai in the Ben-Hur lab. They've been really helpful in some of the work that we've done. So we ran this experiment. And this is now an example of one decay curve with the antibodies present. And it became much faster. And so we did this for a couple different antibodies. We ran multiple trials of all these things. And consistently, the decay time for the antibodies was much, much faster. And so that much more strongly supports the idea of this rolling mechanism on the surface. So again, how does the machine work? It's moving by rolling around on the surface. Alrighty, that's where I'm going to end for today. And then I'd love to address any questions that people have. What have we learned? I've been talking with you about how we developed this approach, this biophysical approach to study viruses. We've talked about sort of chemical requirements for the binding process and how you need these multiple sialic acids, and the involvement of the different sugar groups can influence how that binding might occur, and how we could go about looking at support for a rolling mechanism of mobility. As I said at the beginning, this is like a really small fraction of the things that we do or have done. I was trying to give you a sense of how we would go about doing this sort of thing. I'd be happy to talk about any other sorts of things that we're doing. Right now, we have lots of interesting questions. We're looking at some particular inhibitor drugs that influence certain process of the binding and mobility. We're actually not trying to screen for drugs that block it. We're trying to use them to study particular questions. We're interested in developing membranes that capture more of the complexity of cell membranes. So we're using human red blood cells as a source that we can generate these model membranes ourselves. We're looking a lot of questions about the fusion mechanism. I'll give a plug. If you're interested in some of these questions, Grayson and Sanyukta are two of my thesis students this year. They'll be sharing their thesis talk at the end of this academic year, and they'll be talking about some of these questions in more detail. So you can come and learn more about that if you want. Okay, so that's it. I've pointed out students along the way. These are the current members of my lab and past members. This is one of my favorite pictures from this last summer that we took a lab photo and we decided we were all going to give an awkward thumbs up. So this is everyone's best attempt at an awkward thumbs up. And I love it. Anyhow, that's it. And acknowledgements to our collaborator. I talked today about a little bit some of the stuff we've done with Ben-Hur. We also have collaborations with people at Stanford, and have used some data from people at Harvard for other projects that we've worked on. We, as of today, have funding from the NIH and are grateful for that, and hope that will continue. But also special thanks to the funding that the Williams has given to research students and lab space and many things over the years. And with that, I'd be happy to answer any questions. Yeah, go ahead.
So thank you. This
Yeah.
was great, and I actually understood more than I was expecting, so kudos to you.
Awesome, that was my goal.
But I'm curious about the different shape of the decay functions that you estimated.
Mm-hmm.
Can you explain in a way that I can understand why they would be differently shaped?
Good. So they're both fit to a single exponential, and they do seem to follow that pattern reasonably well. Here you're only capturing kind of the beginning of the decay because we didn't extend the measurement that long. But what you're probably noticing is that the reason for the different decay is that there's a different fraction of the viruses that are unbinding from the surface. And this is something that has actually puzzled us a little bit. So what this is saying in these two experiments, eventually this will level off somewhere down over here, but it might only be 50% of the viruses that actually unbind from the surface, and the rest remain stuck for some extended period of time. With this particular data set, it went down to 70% instead of 50%. There's some variation just between data sets of what fraction end up remaining behind and what fraction don't. So these were just like two examples that kind of represent that. But consistently in the antibody data, there were fewer viruses that unbound. So the ones that did unbind came off much faster, but this is, I think, a real difference, that it did change the amount that unbound. And that's a puzzle that we have to this day. It's on my to-do list of questions to investigate, but one that we haven't gotten to the bottom of. Did that make sense? Okay, awesome, yeah. There was another question back over here, and then we can go around, yeah.
Sure, thanks so much. It was fascinating, and I don't know anything about this, so this may be a very ignorant question, but I'm curious about the way we, in kind of the popular imagination, talk about viruses in connection with what you were saying about the rate of error in replication, and I assume viruses replicate really quickly. And one of the things I've heard about the HIV virus is that it evolves very quickly, and it's one of the reasons why it's been difficult to create a vaccine for it. But we talk about the HIV virus, right, over a span of 40, 50 years, and we talk about smallpox over a span of hundreds and hundreds of years. And they're very small things without a lot of material. So when we're talking about something like a smallpox virus that's changing with each replication, are we talking about the same, meaningfully talking about the same virus that was infecting people in 1500 or something like that? Or do you think about this entirely differently in terms of nomenclature as a scientist from the way I'm thinking about this?
Yeah, so within like a viral species, there will be like an enormous range of genetic variation, and there's different terms or classifications that are used. And the question is usually like, how much does it have to change enough to be considered kind of its own new thing? And usually we're only ever talking about like one virus is not turning into a completely different type of virus over the timescales that we would be talking about. But it might change its abilities or to evade an immune system or something like that. So often how they have historically been categorized would be things like different ways that your immune system will recognize it. If it evolves enough that your immune system has to generate a new set of antibodies against it, then it would be called a new thing. So when we talk about like H5N1 versus H1N1, it's a different type of a protein that an antibody will recognize in a different way. There are constraints for each virus about how quickly it can evolve, what are the, like how much it can actually evolve before it breaks itself, if that makes sense. And this is what explains some of the differences between like HIV and smallpox or SARS-CoV-2 or something like that. That HIV has been particularly difficult because it has a high, not only this, but partly because it has a high mutation rate. And so that within a single individual, you can have many, many different types, and the virus seems to be able to tolerate that reasonably well, so that if you apply a drug, it will kill most of the viruses, but some of them will evolve the ability to just randomly to evade the drug. So that can be something that's a problem. HIV is a particularly nasty case because the cells that it targets are the cells of the immune system itself. And so that is also what generates a set of complications. Most other viruses are targeting other types of cells. Anyhow, that was a little bit rambly, but did that answer kind of your question? Okay, yeah.
Yeah, you mentioned that you think there's multiple binding events required a couple of times. And I'm trying to think about this in my mind. because the picture you've shown is this convex kind of thing, and so I imagine like maybe you could have 3 of those receptors down or something, but the others are curving out to the sides. So like, what do you mean by multiple?
Good, yeah. So this is an example of like, don't confuse the model for reality. This is not at all drawn to scale. So the actual interface would be reasonably large. So I'm trying to remember, we did a back of the envelope calculation for this a while back, but you might have like tens of available proteins at the interface between the virus and the target. So yeah, we're talking about, you might have 20 or 30 that are available that could be binding and unbinding at any given time. So I didn't show any data related to the numbers that what is the evidence of why you need multiple receptors to be bound. Some of that has to deal with, so we have data, other people have looked at these sorts of things, that as you change the concentration of the receptor or maybe the number density of the receptor in the target, if you only need a single binding event, then the dependence of the number density on the numbers of viruses that bind should be relatively linear at the beginning. If I have twice the number of receptors, then I should be able to bind twice as quickly. And instead what we see is what's called the sigmoid. So it's not until you reach some threshold value that you actually start to see binding. And that indicates that you need multiple, because if you're not at a characteristic density, there's just not enough to ever sustain an actual binding event. And so you have to get to a characteristic density before you can actually start to observe anything. So that's one piece of evidence for that. But yeah, again, that was a little bit extra beyond what you asked, but that kind of gives you a picture of how we go about looking at those sorts of things. Yeah, Matt.
Bob, that was so cool. Thanks. 2 very quick questions. One, where do you get your virus?
So the version of Sendai virus that we work with, you can actually buy. So we just buy it from a company and they ship it to us in the mail. There's special packaging and requirements for that in terms of biosafety, right? But that's something we can buy. It's actually, it actually comes, well, you may know it's from Charles River, because it's a mouse virus, and so people who do certain types of mouse research will want this, but we have repurposed it for ourselves. Yeah. My
other question is more sophisticated. It's more, how do you make an informed choice of which membrane receptors to test in the experiments that you described that Amy Lamb did? Like what gives you your basis for making an informed hypothesis for the molecules that you put on the membrane?
Good, yeah. So some of it is like what is available. So what are the types of molecules that have been isolated and characterized? We could go through the process of doing that ourselves, but that would be a very intense effort. So some of it is what is available. A lot of it is also based off of prior literature and trying to understand what has been done. So we knew in advance that the GD1A would work, because people had researched that in the 80s when they were studying Sendai virus at the time. And so then we just began looking at what was available for us that was similar to that would allow us to start generating structural hypotheses. And this was the set of things that we began looking at. But there's more beyond this. We could have tested a lot more, and we just haven't done it yet. Yeah, Chris?
Thank you for the great talk. I know you mentioned that don't confuse the model for reality, but have you found a way of being able to simulate or incorporate the complexity on the surface of membranes? Because I remember seeing a lot of these visuals that have forests of things going on, and in my mind I'm just imagining, would these things ever get in the way of the virus rolling around? Yeah. So this is a question we think about all the time. This is one of the things we're trying to do right now. So this is what has, and actually many people in this field are doing this. So lots of people use, or many researchers, I should say, use model membranes for a whole variety of purposes, not just for virus experiments. And one of the newer waves is to try to create model membranes that have increased complexity in them. Our approach is basically taking a red blood cell, isolating the membrane itself, and then turning that into a version of a model membrane. So we're able to still make a lot of our precise measurements on our viruses, but the membrane now is
a lot more complicated. The price we pay is we lose a lot of control, right? So now that membrane is not something that we can easily swap stuff in and out of. But the hope is that if we have a really complex membrane and a really simple membranes, we can make comparisons between them to get a sense of how much that complexity matters. So that's work we're doing right now. Yeah, stay tuned.
Thank you for the talk. I had a question about the rolling mechanism. So if you used tethered vesicles instead, how does the binding interface and like the rolling mechanism change at all? I was kind of like thinking about like relative sizes of like the vesicle and the virus and like vesicle density, and I was wondering what your thoughts were.
Yeah. So Lisa's actually a member of my lab, so she's talking about tethered vesicles, which are something we haven't even talked about. So tethered vesicles are a different type of target membrane. Let's find an example of one here. Where instead of a flat membrane that's attached to the surface, you actually have a vesicle itself that's been gently tethered to the surface. So instead of a completely planar membrane, let's imagine that we have a version of something that looks kind of like this that never ruptures. So it's just held there, and it's a way for us to look at, among other things, the influence of the curvature of the membrane on the process of the virus interfacing with it. I'd expect that the rolling is still going on, but you've seen examples of this in some of your data. But for sort of more general interest, each of the spots that we observe here, these are what are called diffraction-limited spots. So the virus itself is smaller than the wavelength of the light that we're using to see it, which means that we can never resolve any substructure of the virus. We can't like zoom in more to get more information. There's a fundamental physical limit as to how small or what information we can extract. So this will always look like a blob. And on a tethered vesicle, even if it's moving around on the blob, it's all still within that same diffraction limit. So we wouldn't ever notice it at all. It would just be, it would look to us like it was stuck in place, even though it is in fact moving around.
Thank you.
Yeah.
Well, maybe the last one.
Yeah.
It's a more general question, and it's something that's always fascinated me. You showed examples of different types of viruses, and for example, with the adenovirus, see it looks like an icosahedron.
Yep.
So I've always wondered about the impact of symmetry. How does that impact the ability of viruses to replicate or even the whole infectious cycle?
Good, yeah. So let me hop back. Let's see if I can do this here. Somewhere back here. Let's look at this. So this is shown as a schematic, so it seems like it's idealized. For many of these protein-only viruses, meaning the structural housing is just completely protein, this is not really an exaggeration. They actually look like this under an electron microscope where you can resolve the actual details. So they're very symmetrical and highly organized. And the proteins just self-organize in that particular way. So it's not like this is just a fancy schematic. That actually is more or less how it might look. How that symmetry plays out in terms of the infectious cycle, I think, has not been well understood. There's definitely an idea that they have multiple things pointing out of them, so they have availability to encounter things on different interfaces. But how that exactly plays out, it's possible that somebody's looked at it, but it's not something that I'm as familiar with. Is there anything else I would say about that? Yeah, I don't know. It's kind of an unsatisfactory answer to your question, but it is a real thing that there are very highly symmetrical viruses. One maybe slightly interesting tidbit is, so we study this membrane fusion process that involves like the merger of the membranes together to open up the hole to allow the genetic material to get inside. For viruses that don't have membranes, they have to get their genetic material in another way because there's no membrane to fuse. They have to somehow open up a pore in a different way. And it turns out that it's just like not well understood at all. So for viruses that are not membrane viruses, the entry process, some questions have been figured out a little bit, but how to even study that has been a challenging thing for people. It's one of the questions I actually looked into as possibly studying here. But then I was like, oh, so little is known. I don't even know, like, what's the experimental tool to begin with to start measuring this? And so maybe at some point we'll get back to it. But yeah, anyhow, that was a bit of a tangent, but it gives you a flavor of some of that. OK, should we wrap up? Yeah. Thank you all for coming. It's been great to talk with you. If you have other questions, come on down. I'm happy to chat some more.